From generating simple reflexes to processing complex environments, the architecture of biological information systems takes many forms across the animal kingdom. This chart outlines the structural variations of the nervous system. Early organisms, like the sea sponge, lack a nervous system entirely, while the hydra relies on a nerve net, a decentralized web of separate but connected nerve cells. In radially symmetrical animals like the sea star, individual nerve cells bundle together into distinct fibers. With the emergence of bilateral symmetry in flatworms, neurons cluster into an anterior processing center, forming the earliest iteration of a brain, accompanied by distinct nerve cords. Invertebrates that actively hunt for survival, like the octopus, utilize millions of neurons organized into specialized lobes to achieve high levels of processing complexity. The vertebrate paradigm pushes this centralization further. It relies on a highly specialized brain and a dorsally located spinal cord to form the central nervous system, which then branches out into a peripheral network throughout the body. Throughout biological history, the trajectory of computation drives toward high-density cellular centralization. The human iteration of this network operates at a massive scale, relying on approximately 86 billion individual neurons. However, glial cells, which provide necessary structural and information processing support, actually outnumber neurons in the brain by a factor of 10 to 1. This diagram illustrates the specific morphology of a single neuron. Like most cells, it contains a central factory called the soma, which houses the nucleus and mitochondria. Extending outward from the soma are dendrites. These tree-like structures act as receivers, catching chemical messages from other cells across specialized junctions called synapses. Once received, the signal travels to the axon hillock, the critical junction where the cell integrates incoming signals. The integrated signal then propagates down the axon, a tube-like structure designed to transmit the impulse away from the cell body. At the far end, axon terminals convert that electrical signal back into a chemical release, transmitting the message to the next cell. The physical architecture of a neuron enforces a strict one-way directional flow for all biological information traversing the network. Unlike copper wires that carry electrons, biological signals operate in fluid environments, relying entirely on the movement of charged molecules. This cross-section shows the neuron's lipid bilayer membrane. It acts as a strict physical barrier, separating the cytoplasm inside the cell from the extracellular fluid outside. Because ions cannot cross this barrier freely, the cell utilizes specialized proteins, like the sodium-potassium pump, to actively manage its internal chemical environment. This pump continuously consumes ATP to force 3 sodium ions out of the cell. Simultaneously, it pulls 2 potassium ions back inside. The membrane contains leakage channels for both ions, but because it has far more channels for potassium, those ions diffuse out much faster than sodium can leak back in. This unequal exchange leaves the interior of the cell with a negative charge relative to the outside, establishing a resting membrane potential of approximately -70 millivolts. A resting neuron operates as a tightly wound biological battery, expending continuous energy to maintain this precise state of chemical disequilibrium.
This graph charts an action potential, the brief, intense reversal of that resting charge used to transmit a signal within the neuron. When a chemical stimulus hits the dendrites, positive ions enter, raising the internal voltage. If the stimulus is strong enough, it pushes the cell to a threshold potential of -55 millivolts. Reaching this threshold triggers an absolute response. The neuron will completely fire, independent of the initial stimulus strength. This panel shows the physical mechanism of that firing. Voltage-gated sodium channels fly open, causing positive sodium ions to flood the cell. This rapid depolarization spikes the neuron's internal charge, pushing the membrane potential to roughly +40 millivolts. The instant it peaks, the sodium channels shut and chemically lock. This begins the refractory period. The channels cannot reopen immediately, preventing the electrical signal from traveling backward up the axon. At the same time, voltage-gated potassium channels open, allowing massive amounts of potassium to rush out of the cell, dropping the internal charge back into the negative.
The sheer volume of escaping potassium hyperpolarizes the cell, driving the voltage briefly below the normal resting potential before the leakage channels restore the baseline. What we observe as an electrical impulse in the body is a sequential, highly localized collapse and immediate resetting of this electrochemical gradient. Human motor neurons can have axons up to a meter long. Without modification, a chemical wave would dissipate over that distance. To maintain the signal, the nervous system uses specific glial cells to insulate the wiring. In the central nervous system, a single oligodendrocyte extends outward to wrap segments of multiple distinct axons simultaneously. In the peripheral nervous system, individual Schwann cells dedicate their entire cellular body to wrapping a single segment of one axon. Both methods produce a myelin sheath. By insulating the axon and making tiny exposed gaps called nodes of Ranvier, the signal bypasses the insulated stretches and recharges at the nodes. This specialized glial architecture prevents voltage loss and vastly increases the speed of biological computation. Neurons require a highly regulated environment to maintain their electrochemical gradients and process information. Astrocytes manage this environment. They buffer excess ions in the extracellular fluid, provide necessary nutrients, and offer structural support directly to the synapses. Microglia act as the dedicated immune defenders, scavenging for pathogens and degrading dead cells to keep the network functioning. The computational capacity of a neuron is entirely dependent on this active, multicellular glial support matrix. It was long assumed that the brain's cellular inventory was fixed at birth, that humans possessed a finite, non-renewable supply of neurons. To test this, researchers inject a compound called BRDU, which only incorporates into the DNA of actively dividing cells. This micrograph reveals newly generated cells in an adult rat hippocampus. The red fluorescent labels confirm the presence of BRDU in functioning, newly born neurons. This process of adult neurogenesis is actively modulated by behavior. Learning new tasks and exercising increase the survival rate of these new neurons, while high stress levels suppress it. The nervous system is a structurally diverse, chemically driven, and continuously regenerating biological circuit.